Hi, my name is Michael Kemp. I'm an assistant professor here in the Pharm Tox department at Wright State. And so I thought I would share a little bit about what I do here. So my research interests are in the area of DNA damage and DNA repair, and specifically something we refer to as bulky DNA adducts. A classical example of this is UV wavelengths of sunlight, which can induce thymine dimers in our DNA. We know that these can be mutagenic and lead to things like skin cancer. However, we're exposed to a number of other compounds and carcinogens in the environment and in our diets that also generate bulky DNA adducts that can lead to a wide variety of other types of cancers. So we often think of DNA damage in negative terms. However, we often use DNA damaging agents, such as a classical example is cisplatin, generates adducts on DNA that can help kill cancer cells. And so DNA damage can both cause disease and be used to treat human disease. So in my lab, we're interested in something known as the cellular DNA damage response. We approach this issue in this field in two main areas. One is, again, coming back to UV light in human skin. And our interests are specifically at developing new and better methods for measuring the DNA damage response in human skin, and then to apply these for specific purposes and in specific patient populations. And one of the major projects currently being looked at in the lab is comparing the DNA damage response in young adult skin versus geriatric skin. Since we know older individuals are at higher risk of skin cancer, is this due to abnormal DNA damage responses? A second major focus in the lab is on cancer chemotherapeutics. And in this topic, one of our major interests is in a protein kinase known as ATR that is known to be a global regulator of the DNA damage response in both normal and cancer cells. So we're trying to understand what ATR function is in that context. A more recent project is to look at using drugs that can manipulate and modulate the body's circadian clock to potentially improve the effectiveness and limit the toxicity of DNA damaging anti-cancer drugs. And both of these projects are currently funded by the NIH and the Ohio Cancer Research Associates. So we use a variety of experimental systems in the lab, ranging from in vitro protein biochemistry to quite a bit of human cell culture. Then we also use actually discarded human abdominoplasty skin for a number of types of studies. And when appropriate, we use mouse models. And then we also use human subjects research. And so we will often or occasionally take skin punch biopsies from defined populations of people for our work. My laboratory is located in the Biological Sciences building. I have a number of researchers actively working currently. We have one postdoc, one PhD student, and then currently three students in the Pharm Tox Master's program doing their thesis work with me. I also do some teaching for the department. In the fall semester, I teach a 1 credit hour career planning course where we look at the different types of opportunities that are available for people in this field, and then work to help students try to identify ways to potentially reach that next step once they're done here at Wright State. In the spring, I teach a course entitled Pharmacology and Toxicology of DNA Damaging Agents. Again, this is really focused sort of on my background, research background and my interests. And this course looks at the wide variety of DNA damaging agents we're exposed to, their impacts on cells, how cells respond, and then how these compounds can potentially impact human disease and treatment. With that, my contact information is listed here. I'd be happy to meet one-on-one with people and to discuss my interests in teaching and research in greater detail. Thank you.